In this video, we are going to look about amoebiasis. So, before knowing about amoebiasis, we have to get some knowledge about amoeba. So, it is a primitive unicellular microorganism, which is a protozoa. It has two stages. One is trophocyte and another one is cyst. Trophocyte is an active motile feeding, which moves randomly for its food. And cyst is an inactive resistant infective stage. So it is an inactive dormant stage, and it multiplies by binary fissions. So we know binary fissions in which a cell divided into two, one cell divided into two by fission, binary fission. In this diagram, we can look about how a trophozoite is changed into a cyst. So trophozoite is much bigger and it has a tail tube for a motile. In cyst, it is nothing but it's inactive and small, resistant as well. So most are commensal in humans. It is mostly present normally in human, and it is also present in soil and warm environment. It is also found in low oxygen pressure environment. So these are the place we can find this amoeba. The main causative agent for amoebiasis is Entamoeba Histolytica. Let's have a look about the physiology and structure of it. So as we already know, it has two stages. One is trophocyte and another one is cyst. So in trophocyte stage, it is found in freshly passed stools. If a person affected with amoebiasis is freshly passing a stool, it contains the trophocyte stage of the Entamoeba Histolytica. It is also found in crypts of large intestine, and it is fragile and cannot resist the external environment. So, so only the trophocytes are present in the freshly passed stool. Likewise, cyst is present in formed stools, which is a late. It has taken a long day after the stool is passed. So it has 4 nuclei. It helps in the diagnosis of cyst or trophocyte form. If it has a 4 nuclei, it is a cyst form and resistant to stress. So only the trophozoite form transfer to becomes a cyst form to overcome the external stresses. The transmission of amoebiasis is through contaminated water and food, which is contaminated by Entamoeba Histolytica. The main route of transmission is fecal oral route. An amoebiasis patient who passes the stool and doesn't washes his hands properly, so he is giving a handshake to a normal person. If the normal person doesn't wash his hands and he is having his meal or dinner or something, so he is affected by the amoebiasis, which is transmitted through this route, so fecal oral route. And epidemiology is worldwide distribution. It is present in all over the world, and mainly in India, China, Mexico, Africa, and South America. If you have a clean look of these countries, these are highly populated countries. So it is present in present in highly crowded areas and highly populated countries. It is also present in high tropic and subtropic areas. It is also present in homosexual populations, which is causing cutaneous amoebiasis. Coming up to the pathogenesis of Entamoeba Histolytica. So how the disease is caused. Let's assume a person intakes food which is ingested with Entamoeba Histolytica. So the cyst form of the pathogen is entering into the body. That is ingestion. The cyst passes the stomach. So while passing the stomach, gastric acid stimulates release of trophozoites in duodenum. So while it comes in contact with the gastric acid present in the stomach, it releases the trophocyte. The cyst releases the trophocytes form. The trophocytes dividing causes necrosis in large intestine. So the trophocytes, while dividing, it attaches to the tissues and causes damages the large intestine. Tissue damage due to cytotoxin. The trophocytes also release cytotoxins which cause tissue damage. So this is the pathogenesis of Entamoeba Histolytica. While talking about tissue damage, we have to know how the tissue is getting damaged. So for this, how it is getting damaged means attachment of trophozoites in host cell by galactose inhabitable adherent protein. So the trophozoite attaches to the host cell by GIAP, galactose inhabitable adherent protein. After that, it lyses the colonic epithelial cells, and which results in the alert of our immunity, which brings the neutrophils, lymphocytes, monocytes to these parts, and when they attack the trophozoite, it also affects the other epithelial cells around it. which cause alteration in the cell permeability. So this much of damage to tissue causes the alteration in the cell permeability, which cause irreversible increase in the calcium level in the cell. So which result in the flask shaped ulceration. It is one of the diagnostic factor of amoebiasis, which is a flag shaped ulceration in our large intestine. While speaking about clinical syndrome of amoebiasis, we have two type of amoebiasis. One is intestinal, another one is extraintestinal. So in intestinal, localized tissue destruction in large intestine, and second is abdominal pain due to this localized tissue damage, and crampings and colitis with diarrhea. And other significant symptom is severe bloody stool, which this is due to the damage of tissue and blood flow mixes with the stool of the affected person. In extraintestinal amoebiasis, the trophozoite damages the large intestine tissue and enter into other organs of the body. The most affected organ is liver. So it results in hepatomegaly, which is enlargement of inflammation of liver. Mostly right lobe of the liver is get affected, which result in the inflammation of liver result in the elevation of diaphragm, which is present just above the liver. The other common symptoms is abscess formation, nausea, so due to the damage in the gastrointestinal tract, and fever and chills. The laboratory diagnosis for amoebiasis include the examination of stool sample from these patients. So microscopic examination of stool is very important, which reveals the stool contains cyst form or trophozoite form. So that helps a lot. And serological testing such as lectin binding protein, search for in lectin binding protein or zymodin analysis are done. And genome DNA analysis of this pathogen, which is Entamoeba Histolytica, is searched in the sample collected from the amoebiasis patient. At last, in treatment, prevention and control, in treatment we use a drug like metronidazole, and for asymptomatic patient we use idoquinol and paramycin. There are other variety of drugs, but we are having an example. So the main prevention is chlorination and filtration of water, and having a proper cooked food. So it is transmitted to contaminated food and water. We have to make proper precautions in water and food we take. The main control step for preventing the amoebiasis is maintaining a proper sanitary measure. So like washing our hands regularly, keeping ourselves clean and tidy, keeping our surrounding clean and good. So these are the measures taken to prevent amoebiasis. We used to shake our hands with people to get connected to each other, but besides getting connected, we get contaminated. So I think it's better to follow our Indian style of saying namaste or vanakkam to each other to have a healthy relationship as well as a healthy lifestyle. So if you love this video, like, comment, share, subscribe and hit the bell icon and follow us on the Instagram too. Bye.